Well, hello and good evening, good night, or good morning to everyone, whoever's watching us from wherever of this part of the world. Welcome to this TPCF webinar. It's an honor for us to actually create a community and trying to build up on this community where anyone who's interested in preclinical imaging would be gathering in a single platform and trying to discuss interesting stories and researches on preclinical imaging and its applications. So it's an honor for us today to be the host for Professor G. Alan Johnson, who's a professor of radiology and physics and biomedical engineering at the Duke University. He's also the director of the Duke Center for In Vivo Microscopy, and the title of his topic is Magnetic Resonance Histology. And it's an honor, Professor, to have you here this sort of evening to be able to hear from you talking about magnetic resonance histology. And personally, I'm really interested to see your presentation. Especially, I really sort of value those individuals who are actually trying to move from a holistic sort of in vivo imaging and trying to move it back to the molecular aspect and such as histology. So it's an honor to have you here, and very thank you very much for accepting our invite for this TPCF webinar. And the platform's all yours. Really looking forward to hear your talk.
Thank you, and thank you for giving me the invitation. While I have enjoyed the solitude that has been foisted upon us by the pandemic, I do miss the opportunity of interacting with people. I do miss the opportunity to actually have traveled to Munich. I do miss the beer and the brats, but we'll do what we have to do here. So thank you for having me. And let's start with magnetic resonance histology. So it's worth starting things out by just remembering what has brought so many of us to this place. This is the last sentence in Paul Lauterberg's seminal article for which he got the Nobel Prize. And it is that zoigmatographic techniques, that's what he called imaging back in 1973, should find many useful application in studies of the internal structure, states, and composition. And he ends with the statement of microscopic objects. And I was fortunate to have just received my PhD that year. And so I stumbled into this field of imaging just at an embryonic stage. And it's always helpful to me to look back a little bit and remember the people who have brought us to the state that we are in today. So magnetic resonance histology, what is it? Well, it originated from an engineering approach. In the earliest days of MR, we were working on an early clinical system in which we needed to have something to scan, and it turned out to be one of my dogs. And after dragging this dog back and forth from Durham, North Carolina to Milwaukee to scan several times, I said, gee, I wonder what it would be like to try to scan something different. So we started looking at things that we could scan at high spatial resolution, and we would find chunks of tissue, kidneys and brains, etc., to scan, because as engineers, we just wanted something that was biological. And we became more and more aware of the fact that this method, magnetic resonance microscopy, had some real value for people who were interested in looking at the structure of tissue. Histology is by definition the structure of tissue, and MR histology is that structure seen using magnetic resonance. It provides at least these four elements, and I put them in a kind of a causal mode into descriptions that can cross the boundaries between we in the engineering domain and those now who can take advantage of it. It is non-destructive, so that you can take the same tissue and do other things to it. And you'll see at the end of this talk some very exciting opportunities to start looking with light sheet microscopy. It's 3, 4, 5, 6, 7, 8 dimensions. There are so many dimensions to MR histology that I hope we can reveal as we proceed with the talk. When I speak to my colleagues in neuroscience and those who are not engaged in imaging, they don't want to hear about spin lattice relaxation and spin-spin relaxation time. They would rather hear that this is a sort of equivalent to stains that are used for conventional histology. If we're looking at acetylcholinesterase and NISL, people who do pathology and histology will understand that. So we try to cast our physics concepts into those that people are familiar with. And so I talk about proton stains, and the ones I'll be spending a fair amount of time on today are how we can harvest information from diffusion tensor imaging. And finally, in 1993, when we were doing this, it was inherently digital. Of course, the world is inherently digital now, so that's not that big a deal, but it does provide us some options that are becoming more and more important as we look at studies and look at statistical treatment of studies. So the digital aspect, allowing us to generate histologic atlases, becomes very important. The elements that are most important to getting the signal to noise, sensitivity is sort of like location in real estate. In real estate, the things that people talk about that are most important are location, location, location. And in MR microscopy, the issue is sensitivity. If your voxels are 1,000,000 times smaller than a clinical scan, your signal is a million times weaker. So you have to do some extraordinary things to recover the sensitivity necessary. The three most critical things that we have used are, first of all, operating at a reasonably high field, 9.4 Tesla. This was a magnet I purchased back in 1986. It's still running fine. It's a medium bore, 89 millimeter magnet. We have built our own high Q RF coils. The RF coil that we built is sort of my graduate work was in electron spin resonance. And in electron spin resonance, we would build things called loop gap resonators that are really just solenoid coils. And you see the strategy here is this is a loop of microwave substrate. So there's copper, Teflon, and copper, and it produces a parallel current path, which keeps the resistance very, very low. And now we do make most of these even without solder. So the idea is to get the highest Q resonator you possibly can. About 1995, we stumbled on the idea of what we called active stain. This is not anything new. It's the use of a contrast agent to reduce the spin lattice relaxation time. I suspect that most of the audience here is a little bit familiar with that. I'm assuming the spin lattice relaxation time is the time necessary for the spins to recover between each RF excitation. And for most fixed tissues, most tissues in general, that's on the order of a second or greater. And as you pulse, if you pulse faster than a second, you are not allowing the spins to recover enough, so you don't get much signal. But through the use of a contrast agent, in our case, Prohance, we are able to reduce that spin lattice relaxation time from 1,500, 1,200 milliseconds down to something on the order of 100 milliseconds. So we perfusion fixed tissues with Prohance, and then we have a series of steps that both reduce the T1 without inadvertently reducing the T2. The T2 is the time that it takes for the signal to decay after excitation. In a clinical setting, T1s are, as I said, 1,500 milliseconds, and T2s can be 200, 300 milliseconds. Once you fix tissue, one of the first problems that you encounter is that T2 plummets down to 25 milliseconds. So we've had to do some things to make sure that we get just the right titration of T1 versus T2. There's a little bit of magic in that, but we have mastered the magic to the point where we can do it routinely. And the final thing that is crucial for our activity is a set of gradients. And I'm calling the mouse connectome gradient. For some of you who may be familiar with the human connectome, there is at Leipzig, for example, my close friend Harold Moeller has a machine there, a clinical machine that has gradients on the order of 300 millitesla per meter. These are enormous gradients, taking enormous amounts of electricity. In fact, the human connectome project up at Harvard has their own individual substation. And when they run that magnet, the lights go dim, as I'm told. There's just an enormous amount of current that goes through. It turns out, though, that the current that you need scales as 1/6 power of the radius of the gradient. So we don't need that much current, yet we're able to get gradients for spatial encoding and the diffusion encoding that are 2,500 millitesla per meter. versus about 300 millitesla per meter in the human connectome. So those have been crucial. There are, as best I understand, only 2 of these gradients in the world. I believe one of the other ones is in Germany someplace. I'm afraid I don't know exactly where. The reason these gradients are important to us is because we are doing a lot of our work now with diffusion weighted imaging, diffusion tensor imaging. And again, I'm giving just a brief introduction to it, but in the presence of a normally in the cell, for example, where spins can diffuse randomly in all dimensions, you will get a diffusion that is also random, and the signal will not be dependent upon any gradients that you supply. But if you apply a gradient and look at spins in a projection in the axons that come out of a neuron, those axons constrain the diffusion along the axial dimension. And if I have a gradient that is along that axial dimension, I will get lower diffusion attenuation than if I have a gradient, I will get greater diffusion attenuation than the spins that are diffusing perpendicular to that. So this diffusion anisotropy gives rise to a whole host of opportunities for imaging, which we'll follow up here shortly. And we do that with a classic MR sequence. This is the so-called Stechel-Tanner sequence, named after 2 actually local physicists, 2 gentlemen from North Carolina State University. This is a sequence that was developed in the 60s, and it consists of a 90-degree RF pulse, which excites the spins, brings them down into the transverse plane. Then there is a gradient along any one of hundreds of different directions that encodes the diffusion during this period delta. Then there is a 180-degree pulse that refocuses the diffusing spins, and then another gradient that unwinds the spins. That is, if they've been encoded with some phase in the first gradient, then they are unwound in the second gradient, and that tells you the phase difference, tells you how far they have diffused between the first and second gradients. So since the T2 of our specimens is on the order of 25 to 30 milliseconds, it's incumbent upon us to make this echo time as short as possible so that the signal has not decayed when it's time to acquire it out here when the echo reforms. So it's crucial that the gradients be really, really, really strong so this time delta can be short and the magnitude of the gradient can be very, very strong. And the diffusion weighting is given by the product of the gradient squared and the differences in the diffusion encoding time. So one can see that here. This is a fixed perfused specimen of a mouse brain. And on the left, you see the signal. You see the image when there is no diffusion encoding gradient. Here's all the muscle tissue surrounding the brain. You can see the even some of the detail inside the skull here. Once a diffusion encoding gradient is imposed, all of those tissues in which diffusion is isotropic will be attenuated. The signal will be attenuated. So all of this tissue is attenuated. You see that here. And in the middle one, we have a gradient that is applied along at the angle indicated by the arrow. If we look at the anterior commissure here, you'll see on the left-hand side that the signal is a little darker, and that's because along the axons, the white matter tracks projecting with the anterior commissure here, the spins can diffuse much more readily, so the signal is attenuated. Over on the other side, the gradient is perpendicular to the axonal axis, and there is less attenuation. Just the opposite when the gradient is applied along the anterior commissure parallel to the right-hand side. So this is the fundamental physics behind the diffusion encoding. The math gets a little bit more complicated, and that's because in the idealized physicist world, we have a axon projecting through a voxel. And when we apply the gradient, we get diffusion that tells us something about the anisotropy of that axon. But unfortunately, biology doesn't help us with that. There are, in fact, frequently, practically always, many crossing and merging fibers. In a clinical domain, the voxels that are on the order of a cubic millimeter have in excess of 10 to the 6 axons, neurons in a voxel. So you've got many crossing and merging fibers. And so the way that this is unwound is through some very complicated math that actually is way beyond my pay grade. I've watched and read as the very capable people have been working on this problem for about 10 or 15 years. But it requires that one acquire not just the 6 or 7 different, usually you can do diffusion tensor imaging with a baseline and 6 different angles. And that works as long as you don't have to unwind this crossing fiber problem. But if you want to do connectomics, which is what we're going to talk about in a moment, if you want to look at following tracks throughout the brain, you need to be able to deconvolve this problem. And so that requires that you take not 6, not even 60, but maybe 120, 130. The Human Connectome project right now takes 257, I think, 257 different image sets with the gradients angled at different angles for all of them. So what that results in is very large numbers of specimens. So if we take a single volume and we're looking at, say, 15, 20 microns resolution, we generate very large images with just a single volume. So one 3D volume might be 1.2 gigabytes. As you'll see, sort of at the end of this talk, we get to the point where we're looking at 127 3D volumes, each of which is more than a gigabyte. And you very quickly find yourself with a single image that is 80 to 100, maybe 200 gigabytes for one diffusion encoded volume. And I'll show at the end the addition of light sheet data sets where each light sheet data set is an additional 200 to 300 gigabytes of data. And you'll soon find that you can take for a single specimen a terabyte of data. So we're in realms way beyond what one might normally think about. I hark back to my earliest studies with, I worked on a CT scanner at Duke in 1974. It was the first one we had. It was the second one in the United States. And the data rate was about 80 bytes per second. And the total data set was less than 100 kilobytes. And now we are generating data sets that are a terabyte. It's just an enormous volume, an enormous explosion of data. So we have to have ways to acquire it efficiently, and then ways to deal with it, to handle it efficiently. So we do this with a series of pipelines. The pipeline that I'm showing here is based upon compressed sensing. Compressed sensing is, to me, close to magic. If you are an old-fashioned linear engineer, you think about things, if you don't sample the spatial frequencies that you hope you're looking at, you're not going to see them. But that's not the case with compressed sensing. With compressed sensing, we can sample much less data. In our case, we're sampling about 1/8 of the data we might normally sample for a routine. So the routine studies you would do in a clinical setting, you do a fully sampled data set for practically all routine clinical studies. We're resampling only about 1/8 of the data. Under some circumstances, we're sampling only 1/10 of the data. And if you do so with try to reconstruct it with just the standard analytical technique based upon the Fourier transform, you get images, but the images are of very low quality. You can do a series of transformations into another mathematical domain in which you replace the Fourier transform with a wavelet transform, and that allows you to minimize the structured noise that gets generated with these artifacts and gives you a baseline from which you can then do a more sophisticated reconstruction based upon iterative reconstruction. This is very computationally demanding, where a Fourier transform image can be reconstructed in minutes. The Compressed sensing transform is the data reconstruction takes place over an hour or so, and this requires a significant computational infrastructure we'll see in just a moment. The reconstructed images then, now a four-dimensional array, go through a series of pipelines where they are registered and post-processed, and then they go into another pipeline from which tractography is generated, another pipeline where labels are mapped onto it, and finally, from all of this, you can generate a connectome, which we'll discuss in more detail in just a minute. A connectome is telling me how one part of the brain is connected to another part of the brain. In order to handle the data sets, we must now deal with heavy computational infrastructure. So the way we've been doing this for some years is the individual pipelines where we take information from any one of our three scanners, we throw it into a high-speed switch that can connect to a cluster. The cluster has on the order of 800 different individual processors. There's a local scratch disk that's about 40 terabytes. From this, the data can then go to an archive that is based upon Oracle. And at the moment, our Oracle archive is about 20 terabytes of data. Then it goes into various different computers, each of which is optimized for the image processing pipeline. Several of them are based on so-called grid servers. Grid servers are arrays of GPUs, which can be software configured to handle large image sets that, such as we have here. One of our grid servers has an 8 terabyte SSD and about 1.5 terabytes of memory, so that we can handle all of the data in memory at once to limit the page faulting. So let's move from the technology base to something perhaps a little bit more visually entertaining. That is the use of this in understanding, in all of the cases that I'll have, some elements of neuroscience. These are all mouse brain studies that we've been working on. The first one was a simple question. We asked the question, can we derive image-based phenotypes that look at the circuits of the brain to tell whether individual circuits of the brain are driven by genetics? If we took just 2 or 3 standard strains of animals, could we look at their genotype and predict what certain circuits of the brain might look like? And would they differ? Would they differ from one strain to another? At the time that we were figuring this out, about 2.5, 3 years ago, it was not clear at all that we would be able to delineate differences based upon the diffusion tensor. But that work has come to fruition and was published in NeuroImage in November of this year. These are several of the pieces of eye candy that were generated for the cover. And this is probably representative. The spatial resolution was at 45 microns. The angular resolution was at 46 angles. That is, we took 46 3D volumes with gradients each at individual angles, added 6 baselines. We put these all together in a 4D data set that was 51 volumes, 3D volumes. And then I think we have some problems muting.
Can I ask everyone to mute themselves, please? Mohammed, 831, I think. I think it's all sorted now. Sorry about that.
Okay, can people hear me now? Yeah.
Yep, we can hear you. Thank you.
Okay, so this is a representative image from that study. This is a C57 black mouse, and we're looking at the color fractional anisotropy. In the diffusion jargon, one can look at where diffusion is isotropic, that is the same in all direction, or anisotropic, which has directionality to it. And in white matter structure, there is highly directional diffusivity. So in the corpus callosum here and the fimbria, you can see them outlined because their diffusion anisotropy is quite high. The color in this case is used to indicate the direction of diffusion. So if it's from posterior to anterior, it's colored green. If it's left and right, it's colored red. And if it's in and out of the plane, as you see here, it's colored blue. So the color fractional anisotropy is giving us great information on the anatomical detail. If we look across all of the strains that we studied, this is a C57 black. The D2, which is, these two are parental strains of a series of animals that we are starting to look at with our friends at University of Tennessee. This is the BXD series. This is about 150 different animals whose genotype is well characterized and for whom a catalog of phenotypes exists, a very comprehensive catalog. So the BXD series are F1 strains between the C57 and the DB2, crossed in clever ways so that we have a population with widely different but known genetics. Then we added the cast animal. The cast is another sort of standard mouse. These are all sort of standard mice. And our last one was the BTBR mouse. This is a transgenic mouse that is used as a model for autism. And if you look at these a little bit carefully, in the corpus callosum, for example, subtly, you'll see that the signal is a little brighter here. That's indicating that the fractional anisotropy is a little higher. You see that anatomically, the width of the corpus callosum changes considerably across all of these. And when we get to the BTBR, you'll see that there is an acolossal defect. That is, the corpus callosum does not connect across the midline, nor does the fimbria. So we looked at the males. This is an average of 4 and an average of 4 females. We could look at the differences in strain and the differences in gender. And we were able to see a number of phenotypes, volumes of structures, changes in their anisotropy properties, and so forth. And we were able to do this through the use of a standard parcellization. Back in 2010, we developed a method called an atlas called voxome space. It's named after a charming little seaside town just north of Stockholm, where we were holding a meeting about how to standardize the brain studies that we have undertaken. And that little town was named Foxholm. And we were discussing all sorts of innerwear, outerwear, middleware, underwear. There were a whole bunch of software people there. And at one point in frustration, I said, well, let's just generate a standard atlas. Let's just generate a standard map. And this is sort of equivalent to the Montreal Neurologic Institute's map. It is a parcellation of about 300 structures, 320 structures on both hemispheres of the brain that we can now map onto all of our data in a standardized way. So we have another pipeline that is based upon the advanced normalization tool set. This is used for people doing functional MRI and population studies in the clinical domain. And ANTS allows us to map this parcellation, this set of labels, onto every data set. It's, again, very computationally intensive. We will take a population of 10 or 20 animals, throw them into the cluster, and it will take 3, sometimes 4 days to do a diffeomorphic and affine transforms that are necessary to get these mapped. But it can be done, and it can be done in a routine and standardized fashion. When we do that, then the pipeline allows us to do the following. We look at the individual images. This is a representative slice for one of the images. This is the color fractional anisotropy. Oops. This is the color fractional anisotropy image that I just discussed. If one magnifies and looks at a pixel by pixel level, then on each single voxel, one has up to 120 measurements, 130 measurements here. And one can do a sophisticated deconvolution of the signal to unwind this crossing and merging fibers to come out with populations of fibers. The green are one population, the red are another population of fibers. And then one looks at a region of interest. For example, we have a region here that we can seed. And we seed this region and tracks are generated projecting from this region throughout the brain. And this tractography then is calculated and quantified on a region to region basis. And that gives us data such as this. So in this case, I think we're looking at the LGN. The LGN has been seeded, and I'm slicing through the resulting data where the color code is a log scale color showing the connectivity of everything projected from that LGN. If one does this throughout all 320 volumes, you generate the Connectome. And this was work that was one of my graduate students back in 2015. It was the first high resolution Connectome of the mouse, and it was validated against the standard, which is the retroviral tracers. And the Connectome, as I suggested earlier, is all of the regions of the brain along the y-axis, and then all of the regions of the brain along the x-axis. And then one goes to a specific region of isocortex, for example. We go to one region of the isocortex here, and we say, how strongly is that connected to this region of the isocortex? Or how strongly is this region of the isocortex connected out here to the pallium or to the hindbrain? This is a log scale, a log 10 scale. So the color scale covers 10 orders of magnitude. There's really an enormous dynamic range in this. And we applied this to all of the data, all of the strains that we had, populations of 8 in each strain. And this is the sort of connectome matrix that's generated. It can be viewed sort of like a picture. I mean, it is a picture. And if you step back a little bit, you say, okay, I've got the B6 on the left and the DB2 on the right. Do I see any differences? Well, yeah, maybe I see some lower signal intensity here. That suggests there's less connectivity. Maybe a little bit less connectivity up in these regions from isocortex to whatever it is down in this region. But it's not immediately apparent how different they are. And in fact, from a statistics standpoint, it's particularly challenging, because if one looks at a given region and tries to chart how, if you just subtracted these two, it turns out that there are so many comparisons that could be made that the problem of false discovery raises its head. So from a statistical standpoint, it's really quite a nightmare. So working with some friends up at Johns Hopkins, Kerry Priebe and Josh Fogelstein developed this method where they said, okay, let's look at a region. So this particular, the arrow that I'm pointing to right here is the corpus callosum. That's the corpus callosum. And as we go across the connectome, it shows everything that is connected to that corpus callosum. So it's really an anatomical phenotype of the corpus callosum. And again, the color is indicating how strong the connection is for the corpus callosum to, say, some region of the pallium, or the corpus callosum to some region of the midbrain, and so forth. And if we take this and blow it up, now below it, we can see a little bit more clearly something that kind of looks like a vector, where each color is an element of the vector. And if we look at the corpus callosum versus the BTBR, well, projecting to the other side, from the left side to the right side, we see more blue over here than we see in the B6. And that's indicative of a lower connectivity to the other side. And we'll see that in just a moment. So for every region that we have here, we have a vector that describes the anatomy of that region for all of the data, for all of the animals in the study. And what Kerry and Josh did was develop this multidimensional comparative technique in which those vectors, which are highly dimensional, can be reduced in dimension and mapped to a common space. It is a statistical trick that has been used in many other domains, but has been particularly valuable here. It searches for those dimensions that separate these different entities most effectively. It is in some analog to a principal component analysis, in which now we have the B6 are blue, the D2 are green, the cast are the orange, and the BTBR that are sort of the outlier are the red. And you can see that they cluster in such a fashion that they are separable. And without the BTBR, we see that they are still separable. And one can look at these with a statistical mindset to say, normally, if I have the Bonferroni correction, nothing would be statistically separate. But you can look at this in a different light using this Omni-MANOVA procedure so that you can find a level with statistical confidence that separates all of the regions of interest along the axis here at the bottom. Let's see if I can get rid of this. Where we have a log of the p-value and rank order all of the regions of interest. And when we do so, you see that with the BTBR, we get a separation up here where a large number of these regions of interest are significantly different. What's equally interesting is that without the BTBR, without this one outlier, this one animal, we chose the BTBR because when we set up this study, it was not clear that we would be able to differentiate anything between two very similar strains. But then we were astounded that yes, in fact, we could. So if we separated the BTBR out and just looked at the so-called normal strains, there are something like 150 of the regions of interest that are statistically different. And so we can rank order those and start looking at the tracks. On the top are the several regions of interest. I'm having a little trouble here. All the participants are along the bottom, so I can't see what the bottom is, but I think we've got corpus callosum, internal capsule, and someplace over here, don't know what, let's see what we have here. Yeah, internal capsule, substantia nigra, and fimbria. And you can see that these are all different now. The corpus callosum isn't dramatically different, but the substantia nigra, even between these two very common strains, you can see significant differences in the strength of connectivity. And out at the fimbria, it's particularly profound. If we look at the BTBR, where we have the callosal defect, now we see that these structures are fundamentally different in the BTBR. So we were really quite pleased to see that we could see the differences. If you look at the BTBR, all of the tractography, it becomes exceptionally obvious that the BTBR is fundamentally different. So we see a lot of disorganized tract structure here, particularly back in the cerebellum. You'll look here at on the cortical projections on the side here, and a number of clearly different disorganized tracts in the BTBR, particularly in the motor cortex and sensory cortex. When it comes around again, these projections right here are going into the sensory and motor cortex, and you see that they are diminished up here. And this is consistent with behavioral phenotypes for autism. So let's move to the second study I'd like to share with you is one of looking at opioid exposure. Opioids, as you, I suspect, are aware, are a pandemic almost comparable to our COVID pandemic, particularly in many of the poverty sections of the United States. And so the question is, this is a little bit glib, but what does this do to your brain? How are the opioids affecting the brain? And when we asked this question, I was told by several of the folks that understand neuroscience far more than I, that it was unlikely that we would see any macroscopic effects because most of the opioid interactions are essentially molecular. They are binding to specific sites of the cell, and it would seem unlikely that that would produce any sort of mesoscale operation. The study was set up with 7 and 7, 7 and 6 animals. Animals were giving morphine. The mother was given morphine in a fashion similar to that which a drug addict might be exposed to. These are the exposures and the heroin doses. And this is a representative diffusion weighted image. This is at 36 microns isotropic resolution. You can see layers of cortex. You can see a lot of structure in this. But again, analyzing it gets to be a non-trivial piece of the puzzle. Using the same OmniManova, pushing this data through all of the pipelines that I have been describing, allows us to come up with this representation, which is again separating along the dimensions that the OmniManova defines from the anatomical differences. And we see that the heroin exposed have fundamentally different connectomic projection anatomy. The control are nicely clustered, but these are scattered all over the place. So this is sort of seems intuitively right. We would expect heroin to have a range of exposure. It is surprising and rewarding to see that the heroin is differentiated statistically from the control. But can we take this information and, as we did with the genetic studies, see which specific circuits were impacted? So I'm giving you a little help in interpreting the anatomy. What will follow will be the projections, the connectomic projections for given anatomic structures. And I've superimposed them in this example on the background volume image of the animal. So what you see here is the control and the heroin exposed for cingulate cortex. And you see significant reduction in projections across the midline. You also see significant reductions on the same side. If we look at, so this is cingulate area, this is cingulate, and then now this one was one that puzzled us a little bit. This is looking at the motor cortex. And surprisingly, the heroin exposed has increased the projections to the other side. And there's even increased projections here. That was a little puzzling to us until we realized that one of the hallmarks, one of the behavioral hallmarks of children that have been exposed in utero to heroin is a vastly increased preponderance of children with ADHD. And these are the areas of the motor cortex. The fact that the motor cortex is hyper connected is very consistent with that behavioral defect. So I'm going to conclude my talk with a new dimension, and this is light sheet imaging. I'm going to try to speed it up a little bit so that we can get to the time course here. This was the Nobel Prize in 2018, I think, to Eric Betzig. The idea is that you form a sheet of light and you look at cleared tissue. So tissue clearing has been all the rage. Here is a brain, normal brain, and here is a brain after it is, they are essentially delipidated and then soaked in chemicals that make the index of refraction between extra and intracellular space equal. And when one does so, you can probably just barely see the outline here, the tissue becomes clear. If you put this into a light sheet microscope, where you scan a sheet of light from bottom to top, you can generate three-dimensional images. So what you're seeing here is an image that was acquired in collaboration with folks up at MIT. This is an animal that expresses YFP, yellow fluorescent protein, with Phi1. We scanned it with MR in the skull. We moved the brain out, and they cleared it and scanned it with their light sheet imagers. You're looking at individual neurons. The data set is about 300 gigabytes. You can see individual projections from those neurons. So it's an enormous amount of data. And what they actually do is they get 3 channels at once. So they get You can stain for nu N, you can stain for myelin basic protein, you can stain for cell density, just normal cell density. And we get about a terabyte every time we get a data set back from them. So when we get the data set back, the volume is really, really, really distorted. So what you see in this graphic, the green is the nu N image when we get it back from Life Canvas. The yellow is the same data set after it's been registered to the MR. We use the MR as the template to correct for the swelling and distortion that comes from moving the brain from the skull and then soaking it with all of the tissues. So you see that the distortion is as high as 40%. It turns out it's not really isotropic. It becomes distorted along a couple of different, differentially along the z-axis. So we've generated yet another pipeline that let us register these two data sets together. And you can see them superimposed here, sort of in a macro sense. And then finally, you see the axial diffusivity that I've colored in yellow here. Axial diffusivity is telling us differences in diffusion along the primary axis of diffusion. And in the hippocampus, we see some elegant and simple layers, some elegant differentiation of contrast based upon this contrast mechanism, the axial diffusivity. And here is the Phi1 expression, and here they are merged together. And we believe that this combination of MR and light sheet together opens just an enormous opportunity for understanding brain structure. So to do so now, to propagate this back into our genetics world, we've developed yet another atlas. Our original atlas, Voxome Space, was built back in 2010, and we are now building a new one based upon gradient echoes at 15 micron, diffusion MR at 15 micron, 127 angles. We generate all of these different contrasts down here: axial diffusivity, radial diffusivity, mean diffusivity, fractional anisotropy. We've generated a new set of labels that are consistent with the Allen Brain Atlas. We now generate full connectomes. We get light sheet data at 1.8 micron for 3 stains, nu N, cyto, and MVP. And we are starting a project that was just awarded to us, a significant award from NIH, for us to do 1,000 animals from the BXD series. So to put this in context, the diffusion weighted image in the upper left-hand corner is that of a somewhat aging MR scientist in North Carolina who has probably seen his better days. It is a little bit of atrophy. I'm willing to sign on to that. That's my brain, scanned at a clinical protocol with 2 millimeter voxels. If we compare that to the 15 microns, the spatial resolution in the diffusion weighted image on the mouse on the bottom is 2.4 million times higher. The voxels are 2.4 million times smaller. And if we now add the nu N and the other stains, I can show only one at a time. The nu N has been registered, again, with another pipeline. And if we lay them above each other, you begin to see the exquisite detail that you get from both. This is the dentate gyrus. And look at this subtle little projection of neurons here. You can almost see that you can get it here. And this little projection here, you can resolve it here. Down here, I think this is, I believe we're towards the subiculum. We see 2 and 3, maybe 4 cells. This is about 4 cells thick, and we're able to resolve them here. So the combination of the light sheet and the MR gives us a whole new opportunity. Just briefly, this is the gradient echo. This is the TWI. You see the difference in the 2 contrast. And then this is the fractional anisotropy. You'd like to be able to see all of this at once. So we're doing something similar to the fingerprinting that people have been done. I've assigned color to each different one and then generated the synthetic image. And I'd like to end my talk with this movie that lets you cruise through. We have a grid server, a high performance grid server, that lets us interactively view these even from poor connectivity. I live out in the country and my connection is awful, but I can do all of my work now through the grid server and examine these exquisite data sets in detail. So we're excited about the new opportunities that have come from merging MR with the light sheet. With that, I'd like to pay tribute to the people that do all the real work. I sit out in the country and just look through their work through the computer screen. These are the people that are making it happy. And I thank you for your time. I hope I've made it in time. Thank you so much for your opportunity to speak today.
Thank you very, very much for this amazing talk. I actually really enjoyed it. And it was a new dimension as I sort of expected from the title. And I personally enjoyed a lot and learned a lot as well at the same time. So we're open for questions. Whoever wants to ask any questions, feel free to raise your hand. There's a menu for that, or you can actually write your sort of message in the chat section. So, Professor, we have already a question, and someone called Erfan is asking to see if there's any chance to decrease the size of the data. You mentioned about the gigabytes of data. Is there any chance that you could sort of shrink this?
Say the question once more. I'm sorry, I didn't quite understand.
So the question is there any way that we could sort of decrease the size of the data in terms of the gigabytes? So you mentioned a single voxel would be in sort of, I think I can't remember, it was roughly around that 8 gigabytes or something. And so dealing with this means, is there any chance sort of we could sort of decrease the sort of the gigabyte or the memory of the files in order to work with them?
So there's a couple answers to the question. If you're looking at the whole brain and you want to increase the spatial resolution, you'll increase the volume of the data, of course. And we are working towards that. Right now, we're limited by sensitivity. We have two strategies that we think will allow us to get down to 10 microns, maybe even 5 microns. And the data sets will, if we go down to 5 microns, the data sets will be 10 times larger. So will be a terabyte for every MR data set. The light sheet data sets are 1.8 by 1.8 by 4 microns that we get from live canvas. They're limited by the numerical aperture, and it is possible we have some data right now that has been done at 0.5 microns. And when we do that for a whole brain, a single volume for a single channel would be about 10 terabytes. That's beyond our capacity right now. So we can increase it. We can't handle it right at the moment, but we're working on strategies to do so. The other direction, that is to diminish the size of the data sets so that you can deal with them better. There are a wealth of strategies to do so that rely on the increased computational horsepower from the grid servers. I think the salvation in this is that the computer people are increasing the capacity in both disk memory and computational efficacy faster than we're increasing our data. So we're relying on the computer industry to give us more capacity so that we can get larger and larger data sets. I hope that addresses your question.
Yep, thank you very much. It just shows how complex it is, and literally whatever could be done is actually has happened anyway. I've got a couple of questions myself. So how do you relate sort of magnetic resonance histology, as you mentioned, to a functional MRI? How these two could help each other and how they can sort of complement each other as well?
Yes, the award that we've just gotten from the National Institute of Aging has four major arms, actually. There's the genetics arm that's being run by people at University of Tennessee. They are generating BXD animals in which the 5X FAD gene, the gene that codes, has been implicated in Alzheimer's, is being distributed through animals with widely varying genetic background. A second arm is Marcello Fibo at Gainesville, who is doing functional MRI on some of these animals. And then we receive the animals after the functional MRI. We haven't done this yet, we're just starting, but we will receive those same brains after functional MRI has been done, and we'll be doing the diffusion tensor and light sheet imaging. So our expectation is that just as we had light sheet and MR merged in the same space, within a year, I expect us to be able to superimpose functional MRI for the same animal in the same space with the same light sheet. So another dimension of the data, making it now, I don't know, 4, 5, 6, 7, 8, 10 dimensions. The dimensionality of the data is just getting enormous and terribly exciting as a consequence.
Thank you very much. Actually, another comment I have, or actually maybe it could be a question as well. So you've mentioned about the light sheets and complementing it with sort of MRI. What about, and you've mentioned about the YFP protein, fluorescent protein being used. What about using certain antibodies which are tagged with a fluorescent molecule, or even antibodies which are tagged with a sort of MRI contrast agents such as iron particles? Can we use them at the same time in order to, other than seeing the anatomy and the histology, can we sort of somehow stain them as well in a sense, just like the routine histology or immunohistochemistry that we actually performing these days?
The image on your screen now has been done just with that. This is immunohistochemistry staining with a fluorescent moiety for nu N. And so these are not genetically engineered with YFP. The first animals were. These are now just native strains. After the tissue is cleared, it's stained for nu N, MBP, IBA1. So we get explicit channels for neuronal density, total cell density, and glial density. Life Canvas has a portfolio of about 40 or 50 different immunohistochemistry stains for the AD project, the Alzheimer's disease project. They also have stains for tau and A beta, and we are starting a arm of our project within 3 weeks that would be looking at that. So when we finish with the connectomes and the MR histology, we will get one channel that's showing us A beta, and one channel that's showing us tau, and one channel that's showing us nu N. So there's enormous opportunity there.
Thank you very much. I've got 2 more questions actually. Have you actually studied this sort of Connectome project, or what you're doing, and you described very beautifully, on models of stroke or even behavioral studies, and trying to relate and see if the pattern of the sort of neurons would be different between various different animal models, whether they either been under a sort of a stroke or sort of a behavioral sort of model. Have you studied these 2, or are you planning to study these 2 models?
So it's an excellent question. I just turned 74. I hope to live another 25 years. And in that 25 years, I think my hands will be exceedingly filled with all of these opportunities. We've done some cursory studies of stroke, see some dramatic differences. We have a study that's just being undertaken with colleagues in Germany who are looking at behavioral changes. They have animals that are living in highly enriched environments versus animals in less enriched environments. We'll be starting those studies in June of this year. We have another collaboration that's just started with people up at the Cleveland Clinic, in which we're looking at traumatic brain injury. We've done one study right now with folks in the VA here at Duke, in which we see clear evidence of the optical tracts being altered that we see both in the MR, but more clearly in the MR than we see even in conventional histology. And then we have a behavioral arm of the AD study where we're looking at behavioral phenotypes mapped directly to the structural phenotypes. So I don't think I'm going to run out of models to look at. The question is, can we scale this technology? Remember, we're talking about a single study will generate something on the order of 100 terabytes in a single study. So the challenge to me as a physicist engineer is, can we approach all of these different opportunities in a structured fashion? Do we have the capacity to do it routinely and to do it well? And that's where a lot of our attention has been focused. We think we're there now, and we're starting to apply it in these models.
Thank you very much for this amazing talk. I don't think there's one more question by Fawn saying, can we have live videos of tissues and also analysis on the drug function in the near future, meaning imaging molecular mechanisms? So rather than having so I think probably he is asking, instead of having a still image like what you've shown, can you have a sort of a dynamic, a movie out of these still images?
That's going to be a reach. I don't know how to do that. I think the thing that would, the method that I would argue would be more viable there are some of the nuclear imaging PET or SPECT. The dynamic, these are long scans. The images can take, the standard scan takes 12 hours, and some of these scans take several days. So getting any dynamic information at this spatial resolution is not in the foreseeable future. But I think there are alternative imaging strategies that will do that. And I think the merging of the MR with those alternative strategies will provide some insight.
Thank you very much. With that, I'd like to end the session. And once again, Professor Alan J. Johnson, it was an amazing talk. Really, really enjoyed it. And it sort of opens our horizons into various different sciences and interesting things happening around the world. And it was an honor to have you and to present to us on this amazing magnetic resonance histology. And with that, I would like to end the session, and I would actually ask Leila to actually present the poster for our next session, which is going to be happening in a month's time. So these TPCF webinars are happening on a monthly basis, and our next sort of TPCF webinar would be on immunopet imaging of biomarkers for immune checkpoint blockade therapy by Dr. Natarajan. And so really looking forward to have everyone in our sort of TPCF webinar sort of in next month. And with that, I would like to once again thank Professor G. Alan Johnson for this amazing talk and amazing research, and really looking forward to more interesting data coming from your group as well.
Thank you very much for the opportunity.
Thank you very much.
Have a good day.
You too as well. Thank you.